thank you for joining us today. So let me first introduce you our group. So Tunis R User Group is a volunteer organization. We aim to provide a friendly support network for R enthusiasts in Tunisia and worldwide. This group was founded by three ladies who love programming with R. So Mouna Belaid, business intelligence consultant, Amal Tlili, data science engineering, and Hedia Knani, bioinformatician and data analyst. I joined this amazing group last year, and alongside with Hedia, we are co-organizing bioinformatics workshop using R. As biologists, we really understand the need to learn such programming language to be able to analyze the huge amount of genomic data. To learn more about our journey, we invite you to read our interview with R Consortium just published this week. So our code of conduct, Tunis R User Group is dedicated to be providing a respectful, harassment-free community for everyone. We don't tolerate harassment or bullying of any community member in any form. So please keep this place a welcoming and friendly community. Sorry, So if you missed our previous meetup, we invite you to visit our YouTube channel. You can find all the recording of our previous workshop. And we are happy that our workshop on GWAS organized last year just 5 key views. So, and for today's workshop, we are so happy to have with us Khaled Al-Shamaa, research database manager and Egypt, to teach us the QBMS package for plant breeding analysis pipeline. With a career spanning over 2 decades at ICARDA, he contributed in developing many open source tools, including the R-Shiny framework from RStudio, RGWAS, and many others. He is the author of the QBMS R package, used by scientists to enable their analysis pipeline to query and retrieve phenotypic and genotypic data from different cloud breeding management system. So before we start, we would like to thank our sponsor Epsilon for sponsoring our Zoom and be able to have us today. We invite you to visit their YouTube channel. They have data and machine learning. Thank you for joining us. Please don't hesitate to put your questions Q&R sessions. So thank you again, and the screen is yours, Khaled.
Thank you, Amal. Let me share my screen. Okay, and please let me know if you can see my slides in presentation mode.
Yes.
Yes.
Okay, so thank you for this introduction, and thank you for inviting me to share some introduction about our QBMS R package with colleagues. So my name is Khaled Al-Shamaa, and I'm working as a research database manager and senior analyst at ICARDA based here in Cairo. First of all, once we are talking about breeders and analyzing data, everything starts from data itself. And traditionally, people are used to save their data in a spreadsheet, either Excel or CSV file format or whatever. And we know a lot, there are a lot of challenges and issues with the data saved in that kind of format, because a lot of different sources of the problem, like naming convention, letter case, small letter, capital letter, whatever, then the data entry errors or typo when inserting a data in such a spreadsheet files. The ontology of the data as well, because sometimes even in the spreadsheet, when you said this is a yield, so which kind of yield you are referring to. Is it a grain yield? Is it a biological yield? So all of these issues come up with data in a spreadsheets, as well as the units, scales. If we did not mention very clearly the metadata associated with that spreadsheet, it will be really challenging to distinguish what is the data or what is the type of the data in such a spreadsheet. For example, if we talk about a yield, so distinguish between ton per hectare and kilogram per hectare is an easy task for the domain experts. But if you compare it to the gram per plot, then that will be really challenging to figure out what are the numbers listed in that spreadsheet, as well as the abbreviation used and what are the meaning behind all of that. Still, keeping the data in a spreadsheet format is okay if that one is associated with the required metadata, but moving that data to a more systematic architecture like a database is a step forward in terms of saving data and governance that data. Saving breeding data in databases will give you advantages like data integrity. So we will not talking about a versioning of files or files copied by emails or overwriting some changes done by other colleagues, when all that data will be in a centralized database. Saving a data in a centralized database will enable all users to query that data, not only at the current experiment level or current trial level, but also maybe a multi-locations, a multi-years data that will be much more easier. Because if you think about a spreadsheet files, then you have to work on create a kind of master file and copy paste data from different sources, make sure to align it in the right way. So all of these complications can be waived once you start using a database to save your data. And here I'm talking about data, I mean both phenotypic and genotypic data as well. Access control, using a database system, you can put more control on who can access that data, what kind of privilege. Is it a read only? Is it read write privilege? Or if you would like to put a kind of a review workflow, so once somebody upload the data, somebody else will review that data, accept it or reject it or something like that. Also, it enable breeders and scientists to share that data as an public goods for the on the web or something like that, which is become required nowadays when you would like to publish some analysis or some papers. Depends on that kind of analysis. So saving a data in a database is really a good choice when we think about a long-term storage, rather than saving files in folders and subfolders and external hard disk and all of these complications. Now, most of the databases available out there to work with the breeding data, like BMS, EBS, BreedBase. There is a lot of different databases customized for breeding data. The majority have a kind of two different interfaces to access that data, either through the GUI. So let me take the pointer. So in the GUI case, you will log to that database, and most probably that would be a kind of central database on the web. So you log into that database using a URL, your username and password. Then you have a kind of interface where you put your query, where you browse or scroll over the data. You can download, upload the data from that interface, which is fine for the end user. But now we are talking about another solution when we would like to work with analyze pipeline. What we mean by analyze pipeline, that you would like your data to be extracted from the repository or from the database, do some kind of analysis, then generate a report or an output to build a decision upon that one. That process can be done in two different ways, either manual or let the computers work or the programs work with that kind of workflow. So the GUI is more suited for the human users, while on the other hand, most of these databases provide a kind of API or application programming interface. It's a kind of programming way to communicate between two different entities or two different programs. So the first program will ask to bring or to retrieve the data from the other one, even if they are on the same machine or they are on two different machines. The first program can ask for very specific data by query and build that kind of query, and the other machine will return the required data in what we call, usually for most the APIs, we call it in JSON format or JavaScript objects format. So as I told you, most of the breeding databases like BMS, EBS, BreedBase, and provide or have that kind of API or that kind of facility. The question is how to create that kind of queries and how to retrieve the data in a user-friendly way, in much easier way, because that kind of communication is well format for developers or for programmers. But when we are talking about breeding data, the majority of our end users are the breeders or the scientists. So they don't have that deep experience in how to handle that very technical way to communicate between the programs. So this is why, first of all, there is a kind of consortium or kind of standards there for a breeding API. So it's a kind of agreement between all these components in the breeding ecosystem. Either we are talking about the databases or the devices to take or to collect the data in the fields like Android Fieldbook or even ODK or all of these programs, and the, let me say, a different dashboards or a different analyze pipeline that consuming the data coming from breeding program. So it's a whole ecosystem or a whole community of applications that needs to communicate between each other to collect the required data and make some meaningful results or outputs from that data. What the BrAPI is aiming to is just to come with a kind of agreement, what kind of entities, what kind of verbs, what is the language, how these different components can talk to each other in a way that all of them can understand each other. So there's nothing, let me say, it's not about the software itself, it's about the language, how a different softwares in this breeding ecosystem can communicate between each other. Now, breeders or scientists, their main goal is to have a kind of data-driven decisions. So we have two different scenarios or two different ways. The traditional way, breeders or the scientists will start by collecting a data, saving that data either in a spreadsheet or databases as we agreed. Then that raw data or that input data will feed their analysis using Genostat, using R, using SAS, using SPSS, whatever the statistical package they would like to do to analyze the data, to review or to create that data, until come out with the results that we are going to use it in our technical report or in our presentation, in our dashboard or whatever. This traditional way has an issue with reproducibility because each time you have to redo all these steps manually, because this is how you did all of that. Nowadays, when we are talking about speed breeding facility, when we are talking about genomic selection and all of these new techniques, it come out that we need to take the decision more faster. We don't have a lot of time between harvesting, collecting a data, until do the selection for the next round of advancement. So there is a need to build a kind of workflow, mechanized workflow, or automate workflow that can consume the data directly once it's inserted to the database, do the analysis, and give the final report or output easily. And here where the most of the analysis pipeline, you will find it all over the net, and different institutes, different universities working on developing different pipelines for to serve a different purposes. But in general, all of these systems will start by importing a data or retrieving a data from the source, do a several steps of analysis until you come out with the output or the final output. So once you receive the data, you will do a quality check on that data. You will do a pre-processing, whatever the pre-processing required for your analysis. Then, for example, this is for analyzing a multi-environment trials for wheat or barley or whatever the crop. Then you will start by analyzing a single site analysis for each of these locations, then do a kind of model selection, obtain the prediction. That will go to the next step in that workflow or in that pipeline to do the multi-environment analysis. Then do all kind of auditing and modeling for that one until you reach out the reporting stage. And there you will have a lot of graphs, tables, or whatever. Now, it's all start with data importing, whatever your analysis pipeline is. So is it a genomic selection? Is it a simple single site analysis? It's just to view some general statistics about the data collected so far or whatever. All of these pipelines will start with the step of importing a data or retrieving a data. Now, here we start with the QBMS because mainly when we work on that package or on that kind of functionality, we start by the need. The need for our in-house users was to query and retrieve the data from these databases. So you will find in this QBMS R package, there is no put or delete actions. So you can't upload the data using QBMS. You can't upload the data to these data management systems. You can edit or delete that data out there. So mainly this package to query and retrieve whatever data exists in these databases. Then our end users, where we focus to serve, is the breeders or the scientists. So we are not talking about the software developers. We are not talking about any expert in this kind, because they can use directly the BrAPI standard with the get and post calls or whatever. But for the breeders, they would like to hide all these technical complications under the hood. So we try to use, instead of using the database IDs, they just use the normal name of their trials, of their crops, or whatever. And we will see that in a minute when we jump to the practical part of the session. Then the platform for that QBMS was to serve the R language. So in other words, you can query and retrieve the data using BrAPI standard from the backend database in whatever language you would like, in JavaScript, in Python, in R, in whatever language. But when we build this QBMS package, we focus on the R language because most of our scientists, most of our readers prefer that language to analyze their data. So they will develop their own analyze pipeline using R. And this is why we need that very first step to import, to query and import the data from the databases. So we focus or we build it for our language. So and you can use it inside your R script, in the console, or even in the Shiny, in your Shiny application, or even in the R Markdown reports and documents. The scope, we only focus in this QBMS R package on that one functionality. The analyze pipeline developer need to query and retrieve the data from databases. This is the only task of this package. So there's nothing like do a kind of simple analysis in the package itself. The package is just about to query and retrieve the data from the backend database. The use cases, generally, as I told you, we start from our in-house needs. So and that will that required in so many domains, like in breeding modernization, building a dashboards for the management and all of these tasks. So you can use it wherever your first enter point to the analyze pipeline is the required to get the data from somewhere else. The current status, we have roughly we have 10K downloads since we released it on Crane repository. So mainly, if you think about this QBMS as an BrAPI implementation, it's fair to tell that we are not interested in building or supporting all the BrAPI endpoints, or all the BrAPI actions can be done via that standard, because again, we are focusing on query and retrieve the data. So we are not going to do a kind of implementation for all the BrAPI endpoints. And this is the website. It's already published on a GitHub, and this is the website where you can find more information about the package. So a little bit about the history. Developing this package started in July 2019. At that time, it was, as I told you, it was an in-house needs, and we built it in terms of several functions in the files, and mainly it was to connect to the BMS database in the backend. So that was the version 0.1. Then, after a year, in July 2020, after working with some other colleagues outside of ICARDA, we find out that, okay, this kind of functionality might be useful for other people, not only for our people in-house. So why not provide it as a kind of public good on the internet for whoever may need it? So in July 2020, we make it available on a GitHub under the GPL license. So it's an open source. And that was the version 0.4. Then to make it more reliable, we work with the Crane people to add it to the Crane repository, the official repository of the R language. They need a kind of some steps or extra measures to make sure that we follow the standards in developing that package. And that has been done in October 2021 with version 0.6. Since that time, then we have roughly 10,000 K of downloads. In March 2022, and I believe that was during one of the BrAPI hackathons, we work on adding support to other databases, because you remember we start with BMS, that was our in-house need. Then we said, okay, while other breeding database system providing a kind of support for BrAPI interface, why not start expand the database that we can hook to, or we can query and retrieve the data. So we work with the breed-based people to add a support for that database, and that was implemented or released with version 0.7 in March 2022. Not only that, in May 2022, we work with the gigawatt people, because it's not only about the phenotypic data. It's not about the data you collect in the field, but breeders need also the genotypic data. They need the markers data. And at that time, there was, let me say, two different databases mainly used for these markers data, which is a GIGWA, and I think the other one is GUBI. And again, because of our need, we are implementing GIGWA internally, so we start with GIGWA. So we work with the GIGWA people in CIRAD, in Montpellier, in France, to add a support to that one. And that was implemented and be available in May 2022 with version 0.8. So now using QBMS, you are able to connect to BMS or breed-based and collect the phenotypic data, or query and retrieve phenotypic data, then connect to GIGWA in the other hand to query and retrieve the genotypic data. And this is how you provide all the required phenotypic genotypic data to your analyze pipeline to start any kind of genomic prediction or genomic selection analysis, or even a GWAS or whatever. Now, since that time, we found that so many people start using QBMS, then to have it a kind of production level package, we work for a year on the performance issues in this package. So to make it much more faster and efficient in terms of communicating with the backend servers and packaging the data and send it back to the end user side. So the version 0.9 was only about working on the performance issue, like implementing a kind of caching, zipping the data while transporting the data between server and the client, and so on. And lately, just last month, we added support for another database, breeding database, which is EBS Enterprise breeding system, which is the one breeding management system for CGIR now. And adding a support to EBS add an extra complication at our side in QBMS, because so far connecting to BMS, breed-based, GIGWA, it either, if the backend database doesn't need any kind of authentication, so you can just start querying the data inside that database. But if there is a need to do a kind of authentication, if it is behind the login functionality, then the package until version 1 was able to send the username password from your script to that backend server. But then the industry standard telling that you shouldn't handle the end user username and password by your own code. That will put a lot of risk if there is any kind of flake in your analyze pipeline, or there is any problem in that script that will put the user credential and the user information in risk. Rather than that, we start implementing the OS2 authentication. In other words, in EBS case, if you would like to connect to that database, you said, okay, I'd like to connect to EBS. Then once you run that command to connect, that will open a pop-up window on your browser at their site, asking you to insert your username password out there. You do the login at their end, then they will send back to our script just a token, which will tell your script that, yes, this is a legitimate user, and it has already signed in, and this is the token to continue with query and retrieve the data from our side. So you are out of having any hands on their username and password at all, which is really cool. Now, if you visit the website, there is a lot of information in that website. Especially, I'd like to highlight the articles or the examples available there. There is one example, how to connect to BMS database, to BrAPI, to EBS, to Gigawa, as well as there are a few utility or functions just to access the soil and climate data, but they are not built on BrAPI. So, but because of our end user needs that kind of functionality, so they are available there. But what I'd like to focus on is the breeding management system, either for the phenotypic or genotypic data. So all of that information available out there on the website. Now, okay, for example, I'd like to switch to script that will be a nicer way to. So here, first of all, I have two examples. Let's take this one here. Okay. So first of all, you'll need to install that QBMS package. And here I'll present two examples, one for the phenotypic data and the other one for the genotypic data, just to have a flavor how to handle both of these types of data in QBMS. So first of all, you have to install QBMS on your R environment, and you have two options. Either to install the let me say the release, the official release, which is already on the crane. So you can do install packages in a normal way and just give the name of the package, which is QBMS. Or in case you are working and using something really we are still working on, still not in the official release. So you would like to get an access to the development version, then you can install it from our GitHub repository directly. But be careful, if you work with that GitHub version, most probably it should work, but you have to aware or to understand that this version is under development. So we may add some functionality, change it, remove it, because it's not released yet. So once you install it, then I'll just load the library. The first step is to connect to one specific server that you would like to query and retrieve data from. And we try our best to make that kind of functionality as easy as possible for our end users. Remember, we are talking about breeders, so no need for any technical, like, please tell us the protocol. Is it an HTTP or HTTPS? Please tell us about the port, if there is a port, the path, or all of these information. All what we need, and this is how we develop this functionality. All what we need is just provide us with the URL that you use to log into the GUI for that database. So you are used to work with the sweet potato breed base. So this is the URL where you log in. Then just copy that URL and provide it to this function. Internally, it will split that URL to take out the protocol, the port, the DNS, the path, if there is any path or something like that. Then the second parameter here, telling if there is a need to log in or there is no need to do that kind of login. By default, there is a need for authentication because this is the normal thing for the majority of the databases. But for some of these public database available online who provide the data as a public good, then there is no need to do that authentication. And in that case only, you have to tell, or you have to add this extra parameter telling that no auth is true. So there is no need to do any kind of authentication. Then the third parameter, just to tell what kind of database you are connected to. Is it a BMS? Is it a BrAPI? Is it an EBS? Is it a Gigawa? So because internally, some of the changes need to be done under the hood. So this is all what you have to provide. This is a signature of the server you would like to connect to. So I just run this command. Nothing special happened here. And because there is no need to do an authentication, I'll not call this login BrAPI. But if your BrAPI that you would like to connect to require the username password, then this is how to call it. You say, okay, login BrAPI and provide your username and password if this is a kind of patch script and you would like to run it on the server, or even you can call that login to a BrAPI without providing any parameters. In that case, you will have a pop-up window. It will ask you to insert the username, password, and it will do the authentication with the backend server. So in this case, you will not write the username, password inside the script itself, which is much better scenario. But it depends. Is it going to run as a patch script in the server, so nobody will have access to the console or to the GUI to insert this username password?
Khaled, sorry
yes?
for interrupting. We have two questions.
Yeah,
so
please.
the first question is, does this package cover all types of databases for all CRAPs, or only focuses on a few databases?
Actually, so far we tested with BMS, BrAPI, EBS, Gigawa, and recently we work with the Delta Breed from Breeding Insight in USDA. So these are the tested one. Generally speaking, it can connect to any database that support a BrAPI interface, and there is a way to connect even if it is not already defined to connect to this database. The only requirement that the backend database you would like to connect to is already supporting BrAPI standards.
Okay, so we go, maybe, yeah, we'll respond to our second question. Is there a way to connect QBMS to private databases?
Yeah, sure. You can connect to any database. We use our own, for internal use, for example, we use our own BMS, which is behind the VPN, and it's not accessible for all people outside. And that can be done for sure.
So here, something I didn't understand very well.
Yep, tell me.
In case you put like authentication false, no authentication, so false, you need to provide the username and password, right?
Yes, if that database doesn't allow you to query or to contact that database without providing the username password, then you have to put this one as a false. Then you have to call another function, which is login, to do the login to that database to get the credential that you have the username password that enable you to access this database. Then the rest of the commands will be as they are without any changes. So it depends of if the database itself provides that data without any required to check who you are, or it's a private database where you need to do the login first before you are able to query and retrieve the data from that one.
Thank you. So isn't better, I'm not sure about it, but so putting in your script like your username and password, I'm not sure if it's a good practice. So do you think it's...
No, it's not. It's not. And
Yeah.
this is why I'm telling that you can just omit both of them and call that function like this. In this case, it will have a pop-up window asking you to insert the username, password on fly, communicate with the backend server, and check if you have the credential, send back the token so you can continue with that one. So this is one way. Other way, which is much more better, but it depends if the backend database support it or not, which is login via OAuth2. Login via OAuth2, actually the whole login process will not be done at your analyze pipeline at all, or at your script level at all. Rather than that, it will open a window at their website, at that database website. Then out there, you will do the login. If your username password is correct, then it will generate a kind of token and send only the token to your script side to enable you query and retrieve the data. And you have no idea about the username password that has been used to do that login step at the server side.
That's amazing. Thank you.
Okay, yeah, most welcome. Now, if you notice here that we have a different login function. We have a login function for BrAPI. We have another login function for QBMS, login using OS2, which is used by ABS, another login for GIGWA, and once we shift to that genotypic data example. So unfortunately, because this login mechanism is different from one database to the other database, and it's not included in the BrAPI itself, because authentication is something not related to the breeding data. And each of these databases using a slightly different way or a mechanism to do the login. And this is why we have a different login functions for each of these databases. Now, once I set the server, if there is a need, or configure a server, if there is a need to do the login, then you will log in. If there is no need, then you are ready to go to the next step where you're asking that backend server that you configure it and you already connect to that one. You ask to list what crops are listed there or what crops are available in this backend server. So if I run this command, it sends a request to the backend server, and the server sent me back the response telling that it's a sweet potato. This is the only crop available in this database that I'm connected to. Then I said, okay, it's a sweet potato. Great. Then I'd like to know what kind of programs in that database, because in the backend database, you may have a different programs working on that crop. So here I'm asking to list all the programs available in that database. So you can see here, this is a list of all programs defined or available in that database. Now, what you notice here is that QBMS totally hide the database ID. It's saved internally in the current status of the connection, but for the end user, it only shows what the breeders are familiar with, which is the name of the breeding program. Then once you see the list of the breeding program, if you know already the name of your breeding program or you select from that list, you tell that, okay, this is the list of breeding program, but I'd like to work with Uganda CIP. This is the breeding program that I'd like to open or to enter to that one. So I'll run the set program, and here all what you have to provide is the string, the name of the breeding program. Again, there is no database ID or something like that. Under the hood, all of these technical details are there. But for the end user, you just provide the name of that breeding program. So you are now inside that Uganda CIP. Now, once you are inside Uganda CIP breeding program, you can ask, okay, inside this breeding program, please list all the trials available there. So it just like when you're browsing via the GUI in the website, but here doing that via script, via commands in the R script. So here are all the trials listed in that breeding program. And again, the same mechanism. You list, then you set. You list, then you set. So I see all the trials available, and there's no need to list if you already know the name of the trials. So if you know from the GUI that the trial you are interested in is 2022 season, then directly write the name of that trial. But here I just show the mechanism how to query and retrieve the data. Now,
sorry, we
yes,
have one question. So
sure.
if I want to log in databases other than BrAPI, BMS, or auth, then should I just write login biomart, for example?
Login what?
Biomart.
No, you have to use login, then the name of your database. So login BrAPI, for example, then that will open the login window where you will insert the username password, and you have to use your username password that you used to log into the actual database. So that username, you have to be a user in that database already, and you have to have an access to the GUI in that database. So that is how you can mimic the access to that database, but using a script or a code in here. Does it clear the?
I think so. Thank you.
Okay. Now, we list the trials, then we decide, okay, I need to go inside the 2022 trial. So set the trial with providing the name of that trial. So once you go inside that trial, you can list now the studies. We have to be aware of this terminology because it's a little bit different from one database to other database. In BMS, for example, we call it a study. Then within each study, we have environments. We have where, like when you are working in a multi-environment trial. So in BMS terminology, the higher level, they call it study in their GUI, and the lower level, they call them environments. In EBS, no, we call it a trial. Then inside the trial, we have occurrences. In BrAPI, they call it a trial. Then inside the trial, we have a subfolders. So it's a kind of folder structure where in each of these subfolders, you will have your data. So you have to be aware of the terminology in the database that you are working with, because we're expecting the user or the breeders who are going to use QBMS, he is already familiar with that breeding management system, whatever the breeding management system they are using via the GUI. So you set the trial, this is the higher level. Then inside that trial, you list the studies, which is the locations or the occurrences of I mean, it's the same germplasm list, but we are talking about different environments, either in a different years, in a different locations, or even in a different implementation like irrigated, not irrigated, or zero tillage and conventional tillage or whatever. So these are the environments or the studies inside that trial. Then you can select a specific study, which is listed here. You have the study name and you have the location where that study has been conducted. Then you can select one of these studies and set. So you are now pointing to that exact study, and you are able to start extract data from that one. You can extract the data. What kind of data you can extract from that one? We will start by info or the metadata. So here I'm asking to extract the info of that study. And the beauty of QBMS is that whatever you extracted from that backend database, originally it formulated in JSON format, but inside the QBMS, we convert it into a data frame. So it's now in a very familiar data structure for the breeders or the R programmer, the people who are familiar with R. So here, I can click on that one. This is the description of the study. This is the trial ID. And you will find this is the country name. This is the type of the location. It's a field, not a greenhouse, not a, and this is the longitude, latitude. All of these information are available. So this is a general information or a metadata about the trial itself. It's not at the plot level, it's at the study level. If you would like to extract that data to use it, like map these different studies on a different location on a map or something like that, you can extract that data this way. If you are interested to get the data itself, the spreadsheet, which saved in the backend database, then you have to use this get study data. So once I run this function, and for sure, it depends on the size of the trial, how many records are there, but most probably it will not take that time, especially for the phenotypic data. Once we move to the genotypic data, it's a different story. So here, there is the data I imported or query the data from the backend database and save it in this data frame. You can open it's a usual data frame. It's just like when you do the read CSV file, if you have that data in a CSV file. So you have all the columns with the actual data imported from that backend database, ready in your analyze pipeline or in your R script to start applying ANOVA, regression, REML, whatever analyze you would like to implement on that data. More than that, the beauty of most of these data management systems that whenever you import the traits, your measurements, you already in these databases, you have the ontology built already with these traits. So you can even retrieve the traits ontology. So in other words, whatever you call it GY or whatever retrieved from this database called the GY or plant height or whatever, the definition of that will be already able to extract. So this is the ontology, and you will find here each of these variables. You have all the ontology information about these variables listed in this data set. Is it a numerical? Is it a percentage? Is it a counter? Minimum, maximum? If that link to a specific ontology in the crop ontology, you will have the ID for that one out there. And it depends on the database itself, what kind of metadata that database has about this one. Sometimes you will have a description, how to measure it, expected values, and so on. So all of these will be available to be retrieved. So as you can see here, all of these data, it's just a matter to connect to that backend server, go through selection or browsing. You select the crop, you select the breeding program, then you select the trial, you select the study inside that trial, then you just extract the data from that study. And you can do a kind of loop to extract the data from several studies, from several trials, or even from several programs. The same story is more or less is similar when you are working with genomic data. So this example, I'm going to connect a GEGWA server where SNPs or markers data are saved. So again, I just provide the URL of that database I usually log in where I have my data. I tell that the engine of this one is Gigwa. And again, for this one, it's again, does not require an authentication. So it's a kind of public good, or the data there is just public for testing or something like that. So now I reconfigure QBMS telling, okay, forget about that read base. Now I'd like to connect to this Gigwa. So here I set the new configuration of the server. And again, if there is a need to log in, then you have to use the Gigwa login, either in the interactive mode or in a batch mode. In the interactive mode, you don't provide any username and password. And by doing that, it will pop up a window asking you to insert a username password that will not saved in any file. It will just communicate it directly to the backend server to ensure that you have the credential to access that database. Now, here I'm asking, okay, on that server I just configured, what kind of databases are available there? It will list me all the databases available in here. Then I select one of them. You see the logic. List databases, then set database. Now I'm select one of these databases to access to. Then inside database, I'd like to list the projects. And again, this mimic how you browsing the GUI in Gigwa. So in Gigwa, you start by selecting a database. Within each database, you have several projects. Within each project, you have several runs. So here, I'd like to list all the projects. We have one project inside this database. Even if there is one project, you have to set it. You have to tell QBMS that, okay, I'd like to go inside this specific project. So I go inside it. Then inside it, I'm asking what available runs are there. It tells me that there is only one run named the run one. And here again, I set my current run to be that one. And now I'm able to ask, you know, in the snips or markers data, you have samples and you have markers or your snips. So here I'm asking what kind of samples available there. It will tell me the name of the samples. It query that database for that specific database for that specific project for that specific run. It show me or retrieve the list of samples available there. Then here is the beauty. You can now here. I'd like to retrieve the variance data, the markers data, the marker matrix itself. Now I'm telling that, okay, we have some filtering parameters like telling, okay, the maximum missing ratio is 20%. The minimum minor allele frequency is 5%. And just here, for example, because, you know, marker data can be huge and it can take a long time to retrieve that data. So just for this example, I restrict telling that, okay, I'd like to import a data or to query data that start from the position 100,000 until the position 500,000. Just a very small chunk of the data just to demonstrate how that will work. And even you might not interested to import a data for all the samples. You may are interested in a specific sample, so you can provide a list of samples you are interested in. If you did not provide anything here, it will import for all samples and the same for all other filtering criteria. So here, by running this one, and because this might take a longer time to import the data, so you'll have a kind of progress bar until the data imported. And you can find here, we have something like 2,600 markers. And if you open it, it's just in a normal format that usually you work with in the HapMap files. So you have the name of the markers, you have the alleles with the major allele frequency / by the minor allele frequency. Then you have the name of the chromosome, the position in base pair. Then you have your samples or the individual that you have it there. And QBMS query and retrieve the data in a numeric format, where 0 referring to the reference allele, 2 referring to the minor allele, and 1 in case you have heterozygous cases. And in A, it's clear that for not available data. So
sorry,
yeah.
I will
Yes.
interrupt with one question. So is it possible to extract the data for all studies in a trial in one go, or do we have to do that one by one?
For all studies from the same trial, there is there is a function. We call that function multi-environment trial, and let me show you here. For example, this is a base example. Here it is. So if you would like to import all studies in the same trial, so in other words, you have exactly the same germplasm list for all of these studies, but it's only about different trials. So you can call the get trial data instead of get study data. So get study data, it means for one specific study. Get trial data, it will go internally in a loop to import the data from all the studies. But if you are interested to get all trials data for a specific breeding program, then you have to do it. You have to do it in your R script. You have to do a kind of loop where you list all the breeding program, then you go inside that loop to import for each one. So because in the package itself, we would like to make it a kind of the main entities that require. So yes, we agree that many breeders might be interested, especially for the multi-environment trial, might be interested to import all studies data belong to the same trial. So we have a functionality for that. But all trials for a specific program, there is no built-in function in QBMS to do that task. So you have to do it in a kind of loop or something like that in your script. At the end of the day, you have your R script and you can do the mechanism you would like to.
Thank you.
Okay, so let me back here. So we went through an example. So, and you can see here, if you ask to log in for some of these databases where the login is required, without providing the username password, you will have a pop-up window where you insert your username, password, then click on OK button to check the credential with the backend server and give you the authentication to start query and retrieve the data from the backend. Now, what is coming next? Once we work with the genomic data, we figure out that there is a bottleneck when we importing a markers data, especially when we are talking about mid-density and high-density markers data, that it takes really a long time to retrieve all that data. If you have a case where you have 100 of samples and 1,000 of markers, then that might take an hours to retrieve the data, which is not practical in many scenarios. So this is why we work with the GIGWA people in CIRAT on improving the efficiency of how to import or retrieve the data from the backend genotypic data server. Actually, in BrAPI standard, there is a kind of endpoint where these allele matrix data can be transferred in a format of a matrix instead of a list of pages, which is much more efficient way to send the data in 2D instead of 1D. But that kind of BrAPI endpoint was not available in GEGWA until version 2.6, which released in last May 2023. So once they implemented in their backend database, and this is why how I back to the question of the colleague who asked, is all the breeding database supporting this one? Actually, it's a kind of relationship between us in developing QBMS and the database developers. So we need to ensure the required integration from both sides to do that kind of connection and communicating. So once GEGWA people develop and add support for that allele matrix, we work on implementing this functionality also at the side of our QBMS during the last BrAPI hackathon just a couple of weeks ago in Germany. And here is a screenshot of to compare between the existing way of importing GEGWA get variants and the new way, which will be released with version 1.2. And even the people who are interested to test it, they can download the development version of QBMS, not the one on the screen. So you can see here, to import the data, it takes, for example, in the old way, 1.1 minutes, but in the new way, it just takes 5 seconds to import exactly the same data set. And we ensure that we check that the integrity or the completeness of the data. So it's really amazing how that can make it much more faster, which is putting QBMS in a great position to be implemented in several genomic selection and GWAS analyzed pipeline, because it's now it's more practical to import the data from the backend database directly via API rather than download it in BCF files or in HapMap files or whatever files, and let your script to open that file and read the data from the file. The other thing that we are working on for the next version of QBMS is to have a function that can call a kind of shiny widget. So in other words, we look to our end users, and they mainly, even if we have all of these steps or function calls one by one that you connect to the database, then once you connect to the database, you list the curves, then you select the curve, you list the programs, you select the programs, and so on. All of that is available now in QBMS via commands or functions. But most of our users, they would like to, it just like when you read CSV file, I'd like to call one function, I have a pop-up window, do the selection, and the data will be retrieved as a data frame. That's it. The end user is not really interested to see all of these steps as codes or calls for functions and at their end. So this is why we are currently working on build a kind of shiny widget that it's a one single function call. It will pop up a window and it will show you something like this, that what is the type of your database? Is it a breed base, BMS, EBS, whatever? You select the type of the database, then the second question, okay, give us the URL. This is the URL. Click on save. That will change the WYSIWYG or what you see into the authentication step, which is the next step. And you have two options. Either you know that this database does not require the kind of authentication, so you check that a kind of checkbox telling that no, there is no authentication in this one. Or if there is an authentication, you uncheck that one, then it will ask you for the username, password. You insert it, then click on login. Then it will start by showing, okay, you logged into this database. It will start by showing the list of breeding program as a combo box, a drop-down list. You open that one, select one of these breeding program, then the next list will be automatically pop up with all the trials inside the selected breeding program. Once you select one of these trials, then you are able to select one or multi studies in that trial. Then click on load. At the end of the whole process, that function will return a data frame. So in other words, even currently you can do all of these steps using programming calls for a different function in QBMS. We would like to provide a kind of wizard. So the user will only, okay, I'd like to import a data from using BrAPI. Then all of that will be hidden under the code. You will have only a pop-up window. So that will be a very nice add-on once we add it to the system.
So before, moving forward, so we have two questions.
Yep.
Does the QBMS package applicable on different types of omics data, such as proteomics or transcriptomics data?
I did not get the question.
Okay, sorry. So the question is if the package is applicable on different types of omics data, such as proteomics or transcriptomics. It means that maybe will you add in the future, you know, it's like any kind of omics data to the package?
Currently, it's not in our plan because, as I told you, this package is driven by our user needs. So if we find in the community of users more interest in that kind of data, then we are willing to add that kind of functionality as far as they are supported in the BrAPI standard, BrAPI breeding API standard. If that kind of data is not already standardized using BrAPI, then QBMS can't do that kind of implementing for the functionality. So you see, there are several levels. The standardization, first of all, so that kind of data has a standard in BrAPI. Then in the backend database, they implement that standard because standard itself, it's something like a configuration or a template that, okay, you can send or you can query the data using this kind of verbs, this kind of query. But it doesn't tell the database provider that you have to implement it. It's an optional for each database. But again, it's a kind of ecosystem, and it's a win-win situation. If the database developer support more and more endpoints of BrAPI, then that will make it more accessible to the community and make it more able to integrate with the different tools in this community. Then after the database provider develop or implement that kind of API, then it's our turn to look to the community, and they said, okay, we are interested to get the data in that format or that type of data. Then we have to work on adding that one. So this is the how things evolve in QBMS.
Okay, so to summarize, like, other than phenotypic and genotypic data, if it's not in BrAPI, so it will be, it's
out
not
of scope.
possible. Yeah. Okay, thank you very much. So we have another question. Yep. When is the GUI version Shiny coming? Is there a release date?
Not really. We expecting that within the coming 2 to 3 months. Usually the rounds between releases is something like 4 or 5 months or something like that. For currently the GIGWA calls to import a genomic data is pretty ready. So I believe that we will work on this shiny widget, and it might take a couple of months. So maybe you can expect it by end of June, start of July, most probably. There is no fixed date yet.
Thank you.
Now, where that QBMS already used or listed? Actually, we have several use cases where people or developer inform us that they are using QBMS in their products. For example, as I told you, we start with our in-house need. So we have a several analyze pipeline where we need to import the data from backend database. One of them is the fake analyze pipeline. So this is one example at ICARDA. Another example is what we are working on currently, which is the CGIR breeding analytics pipeline. And that kind of analyze pipeline, again, it needs to import the data first before implement any kind of modules to analyze the data. And we implement QBMS as an entry point to import the data. My friend Johan from SIAT, also the developer of Mr. Bean web application to analyze the data, also implement QBMS internally. So in Mr. Bean, you are able to upload a file just like a normal CSV file, or you are able to connect to a BrAPI endpoint, either BrAPI or BMS, to connect that backend server and import data. I'd like to highlight the one implementation or one use case by Francesco Agosto, which is actually not an implementation of for the QBMS itself, but rather than that, he built a kind of application or a kind of service upon that QBMS. So it's a kind of shell to the QBMS, enable the end user to connect to a different database at the same time, which is a very, very nice work. You will find a link for Augusto work in the homepage of the QBMS. And as I told you, while we are working on implementing a several database, it's driven by the community needs or the user needs. And recently, we have some collaboration with breeding insight people in Delta Breed, which is another breeding management system, to integrate it with the QBMS, or to ensure that there is enough BrAPI endpoints required to integrate QBMS to query and retrieve that kind of database. So we have a very positive relation with the many of the database developers to ensure that the ability to connect between these different systems can be aligned and can be achieved smoothly in an easy way. And QBMS already listed in several lists, like in excellence in breeding, BrAPI compatible softwares, CG Space and MIL. A lot of people mentioned that one in there. And that's it.
Thank you very much. So do we have more questions, please?
Great. Either it's too clear or it's...
So I have a question. Is there a way
Yeah,
to
sure.
collaborate in your Shiny app? So if someone wants like to collaborate, yeah, for the like Shiny app implementation?
Yeah, sure. You know, it's an open source community, so everybody is welcome to give us a help hand in doing this or in implementing this one. Actually, if you check the GitHub repository, we have few other developers from all around the world. Even somebody said, okay, I need that functionality. Then I tell, okay, it's nice to have, but I don't have enough time to do it. I can't do it. Then, okay, develop it. Make a pull request, check it, merge it, and it will be part of the package. And this is how I first introduced to Johan, because it started by, okay, I need that functionality and I don't have enough time. He do that kind of adding these functionality. And I believe this is the mindset for the majority of the open source projects. It's a kind of collaboration between different people worldwide who are interested in improving or enhancing for their own needs, for the community need, and I deeply believe in that.
Yeah, the other question for the Shiny app, you know, it's like maybe it's like you can add like transcriptomic data, but to the Shiny app, not to the package.
Yeah, you still able, because think about it just like a one source of the data. So you can import the data from CSV, you can import it from BrAPI, and you can import it from other sources like any other technique. So it's just one extra way, make it more easy to communicate with the backend databases.
Yeah, thank you. So do we have more questions? I'm not sure if we have more questions. So everyone is saying thank you. Please feel free to ask question if you have any, if it's not clear. So we have a recording of the workshop, and yeah, we will send you a link of the to our YouTube channel once it will be ready on YouTube. And I think also we will send you again, you know, all the links related, or all, it's like the package, the teaching materials, everything, like the slides. If you have still any question, or if you have like any bug, you know, don't hesitate to let us know, and we will do our best, you know, to help you with it, to solve it. So I think if we don't have more questions, so thank you again, Khaled, for this amazing workshop. Thank you everyone for,
thank you, Khaled.
Yeah,
Thank
you. And I let Amalia say like thank you and goodbye to everyone.
Thank you, everyone. Thank you, Khaled, for this informative workshop. So we looking for other collaboration in future, maybe other
Inshallah.
workshop on other aspects. Yeah, thank you so much. Thank you, everyone, for
Have a nice
joining
day.
us today. Thank you.
Bye-bye.
Thank you. Bye-bye.